designed specifically for thin films and then detected. And again, one of the things that's in process is to place the ESI and SPR instrument in that sample spot to collect all of that data at the same time. So one of the things in terms of scattering experiments that's actually quite useful when you think about neutrons is that neutrons scatter off atoms and X-rays scatter off electrons. And if you're scattering off atoms, one of the advantages of the neutrons is that different atoms can be matched to different refractive indexes, and so you can actually contrast match your samples. So what do we mean by that? Basically, you can match, this is sort of a visible light example, so if you look at this rod, okay, you can see it in air, but you can't see it in the silicon oil because the light refractive index of the oil and the glass are the same. And the same thing's true with the neutron measurements. There's mixtures of D2O and H2O that can be made to match. And that's Susan Kruger, who's one of our NCNR scientists who helps primarily with stance measurements, but this is her slide. So basically, if you're looking at contrast variation, what we can do in a complex assembly of a lipid protein system is we can match one of the components and basically blank it from our experiment. So you can match the protein or you can match the lipids, and so you can look at scattering selectively from one component of your sample. The other thing you can do is you can deuterate your samples, which again allows you to look directly at the deuterated sample and match it against the protonated components of your assembly. And that turns out to be very useful again for membrane proteins, where you can have deuterated lipids, deuterated proteins, and then various levels of contrast matches where the protein contrast match